He was the department chair and professor of chemistry at the University of Kentucky. He has lectured worldwide and testified before congressional committees in the Institute of Medicine on mercury toxicity and neurological disease. Dr. Haley has dedicated the past 6 years of his research efforts towards developing a safe and effective chelator for mercury, lead, cadmium, and other toxic metals. Please join me in welcoming Dr. Boyd Haley.
What we're going to talk about, when I say a single scientist view, because I don't want to blame anybody else for my aberrant knowledge, etc. But we have to address some of the problems that are existing in America today, and you can read this as well as I can. But medicine has numerous illnesses of no known etiology. We don't know what causes them. What we know is that they're modern man diseases. They didn't exist before the advent of amalgam fillings or the exposure of mercury to people for a long time. And there's a list of them here: Alzheimer's disease, multiple sclerosis, and ALS. And in spite of the scientific evidence indicating an involvement of heavy metal toxicity in the etiology of some of these illnesses, it is still widely disputed that exposure to such heavy metals is involved in the causation of many of these illnesses. And what I'm going to show you to start with is a talk that I gave. I think David Kennedy told me it started in 1989 when I first came into this academy as what I call an Ivy League or Ivy Tower type scientist doing research. And I was highly funded by NIH on a technology called photoaffinity labeling, which requires making compounds that explode when you hit them with light and label certain proteins. Very elegant, very good at diagnosing changes in the energy regulating proteins in the body. And it was widely accepted by NIH. And you're going to realize that when I start showing that using this technology, which NIH funded for over 25 years, when I started showing that it could measure the effect of mercury on neurological tissues and showed that the effect could be mimicked by mercury, all of a sudden it wasn't such good research anymore. And so like a lot of people, the minute you start telling the government that some of the technologies and the things that they propose to support, such as dental amalgams, mercury in vaccines, is not good, you're not going to be funded. You're not going to be in there very long. So we're going to look at some of the things here, and you can read this. This gets pretty thing. And so this talk will be presented in the following sections. First, I'm going to give you the published data that's showing that mercury at very low levels in all the appropriate test systems can reproduce all the accepted pathological markers and abnormal biochemistries of Alzheimer's disease in AD brain. Facts totally ignored by medical science and regulatory agencies today. You can make every diagnostic hallmark with very, very low levels of mercury. We're going to show the science. This is not all done by me. I just happen to be the first one. And so what this tells you is that the change that we need in this country is not in the science. The science is there. The speaker before me, Dr. John New, talked about the 2006 paper from the FDA saying that dental amalgams were proven totally safe. I was on that. I testified. I put data in there showing that mercury was toxic. I don't recall ever reading a paper saying that dental amalgams was safe scientifically. I read a lot of papers by the American Dental Association and dentists associated with them saying it was safe, but there were no safe papers. There has never been a paper published that I would look at that I can recall with all my experience that shows that mercury vapor is safe at any level to anybody. That paper was just because the FDA did not want to step on the toes of the American Dental Association and say your amalgams are not toxic. Everything that's come out since then has said it is. And now if you remember, the United Nations has a proposal. They have suggested, and they have the Minamata Bay Treaty, which they're saying we're going to phase out using dental amalgams throughout the world. If they're so safe and so effective, why are the rest of the countries in the world saying they shouldn't be there? So we're going to talk about that first, to show you some of the stuff and how can they possibly ignore that. In a second, we're going to talk about some of the biomedical problems of treating heavy metal toxicity in the central nervous system and how we have to address this. And the third is the latest evidence that NBMI, a hydrophobic metal chelator that I have designed and developed, how it is capable of entering the central nervous system, go into the brain, bind a toxic metal, eliminate the oxidative stress in this animal model, and prevent the animal from becoming demented and lose the use of its limbs. It's, I think, a very major development in this regard. Okay, this is it came from Dr. Michael Ashner's paper. Ashner is on our biological SDS, the Scientific Advisory Board for this committee, and he published this, and it's really very true. I've added a few things to it, and this is the inhalation of mercury vapor coming off of your dental amalgams or off of any source. 80% is absorbed in the lungs, and then in the lungs it can get to the blood, and there's an enzyme there called catalase. Catalase convert mercury vapor, uncharged, into Hg2+. At the same time, Hg vapor is freely permeable to all the blood-brain barrier. It gets into the brain. Once it gets into the brain, it gets, again, I've lost my pointer, converted by catalase and other enzymes to Hg2+, and it gets trapped in the brain. Now, in the brain, as the previous person talking to us showed, what does mercury do in the brain? Mercury is not a redox metal. Mercury cannot make hydroxy-free radicals that causes oxidative stress. But mercury displaces iron off of these sites, and that iron is very catalytic in producing hydroxy-free radicals. So when you say, how does mercury, because I had this fight early on in my career, that people saying mercury cannot cause oxidative stress because it cannot donate electrons to hydroxy radicals, making them reactive. Well, it doesn't. It displaces the iron, inhibits the iron transport process, as the previous speaker talked about, and I've talked about many times here in the mitochondria. But that iron that's released starts generating hydroxy-free radicals and causes the oxidative stress that's associated with the disease progression that we call Alzheimer's disease. And you can read all this that we have there, and so what we can see, there's a total tie-in of mercury causing oxidative stress that causes the damage in Alzheimer's disease. But it goes deeper than that. And we're going to talk about mercury exposure, and I presented this to the Academy in my first invitation to talk to these people when I thought they were all toxicologists. I said it was in 1993. David Kennedy says, no, it was 4 years earlier in 1989. That means that was 27 years ago, and at that time, I didn't know that there was mercury coming off of dental amalgams. I could not believe, when I first came to this academy, that people talked about mercury coming out. Somebody put mercury releasing products in your mouth. That's a height of stupidity to a biochemist, the height of stupidity. And it exists today. And you don't change their mind by just telling them about it, because we have presented this data to them. And that 2006 white paper was written by people who were either total idiots or they're crooks and criminals. Because there were no papers... There were no papers at that time, and I don't know of any yet today, that show that you can expose any living organism to mercury without causing significant damage. And that's what they said. There's no proof that mercury vapor or anything like that causes such a deal. So what we're going to talk about is how this ties into Alzheimer's disease, and the mercury. You have an etiology. You have to say, it makes sense. There are a lot of things that can cause tubulin to depolymerize, like you see in Alzheimer's disease brain, but you're not exposed to them. But mercury vapor, if you got a mouthful of dental amalgams, as Dr. Yu just pointed out, you're taking in mercury vapor on a regular basis over a long period of time, many tens of years. And mercury vapor causes this process, the tubulin, to abnormally polymerize. And tubulin is the major protein in the brain, and it's used to hold the axons and the synapses, everything in a proper orientation by a polymerization process. And this is a cartoon of this, and it shows an axon, and one of the places where let's see if I can get it up here. This is tubulin right here, these little globules here. They line up and they make a railroad track that motors climb on like this and carry neurotransmitters to the end of down the axon. If you go up here, it goes up here, it goes out here, and it's released to cause a nerve action potential that you need to do if you're going to think or walk or do anything. So tubulin is fundamentally very critical in the brain. And we're going to show you that mercury causes this to break down very quickly and very rapidly. Now, if you do a different cartoon to explain things, you're going to see where do some of the biological markers for Alzheimer's disease come. Neurofibrillary tangles. They're formed from the neurofilament that holds the direction of the tubulin. The tubulin is up here surrounding this, and if you take this apart, and we'll show you the film, we're not going to show the film, but we're going to talk about the film from Calgary, which shows that if you take off the tubulin, all of a sudden these neurofilaments form into a tangle, which is called NFTs, is the major diagnostic hallmark of Alzheimer's disease. So mercury can induce the formation of neurofibrillary tangles. The microtubulin, we're going to show you, is disrupted in almost all of the Alzheimer's disease brain, totally non-functional. If you take these people after they die and you take their brain, compare them to normal people or people who aren't demented or have Pick's disease or some other form, their tubulin is normal. But in the Alzheimer's disease brain, it's not, and you can mimic this by adding mercury. But also point out that there's a tau protein that connects the neurofilaments to tubulin. The other diagnostic hallmark of Alzheimer's disease is the hyperphosphorylation of protein tau, all in the same system. So we're going to talk about these things and show that mercury does this. And so what I was really well known for before I got to know you guys, and even after I got to know you guys, was making photoaffinity probes. I made radioactive compounds that exploded when you hit them with light and would bind to wherever they were sitting on the thing. So we could look at GTP binding, and to make a long story short, this A to zero GTP, radioactively labeled, would support and mimic GTP in the polymerization of tubulin, and we published all this in such journals as the Proceedings of the National Academy of Science in 1977, way before I ever came here. We showed it was on the east side of the beta subunit, which is the one we're looking at, and it's hydrolyzed to cause the tubulin to form into a structure that stabilizes the neurons axon, and we had used it to identify the base binding domain of GTP, of tubulin. And so here was the study we did. Now how this happened is I had a student who learned to do photoaffinity labeling in my laboratory. She graduated and she took a postdoc at the Aging Center at the University of Kentucky, where there was a Dr. Marksberry that was very big into Alzheimer's disease studies. And she came back and she started telling me the problems they had with it. Nobody knows what causes it. And so I said, well, let's look at it. Let's see if we can look and see if there's a change in the nucleotide binding proteins in a disease versus a non-disease state. And this is one of the first autoradiograms we did, and I can't explain this to you because you don't know enough biochemistry, but just suffice it and trust me to say it got published in the PNAS, so it was probably pretty acceptable. And what we saw, if we took control brain, and this is beta tubulin that binds GTP, and we can label that, normal brains, every one of them have a very high level of labeling. If you get over to the demented brains, and we didn't know what dementia these people had at the time we did the study, Alzheimer's, Pick's, and other forms of dementia. But you can see that some of them didn't have any tubulin at all, and that was a major protein in the body, and most of them were down quite a bit. Some were normal. And what we found out that those that had the normal level turned out to be Pick's. They didn't have neurofibrillary tangles because their tubulin was okay, and their neurofilaments were covered and protected. So we did this, and this turned out to be right. We published this, and it's been highly regarded, and everyone said he's right. No one has ever said I was wrong about the tubulin in Alzheimer's disease brain not being normal. That's fine. That was good for study. I got a 5-year grant from the NIH to study this, and made everybody happy. So then we started looking, we did some more research on this, and we found out both the alpha and beta subunits of tubulin are found in equal amounts in the brains of Alzheimer's disease and normal people. So that the protein is not disappearing. The protein is there, it just doesn't work. So that means something has to come in and block the functionality of that protein. And then I read a paper. This was done on 500 sets of identical twins from World War II. And what they said, because everybody says Alzheimer's is genetic, it is a genetic susceptibility to a toxin, but it's not genetic because you could find identical twins, one that would get Alzheimer's disease and the other one did not. However, there was a tendency for both of them to get it if one of them had it. And that means that they're susceptible. But you have to have a toxic exposure to get it. That's what the papers came out and convinced. And so we checked the Alzheimer's brain and the control brain, and you can see that the tubulin is there. It's just something has happened to it. I mean, it wasn't not made. It wasn't chewed up by proteases or anything. The tubulin is available in AD brain and is there without any problem at all. But then we did a very simple study, and anybody sitting in here that has a centrifuge that's a scientist can repeat this study, and it has been repeated, and I'm right. The tubulin in a human brain is mainly in the soluble fraction. If you take a brain, you grind it up, and you spin it down, and then run it on a gel, if you have a normal brain, most of the tubulin will be in the soluble fraction, floating in the water above the pellet. In Alzheimer's disease, there's almost none in the soluble pellet. In other words, it's abnormally polymerized. Now, what can cause abnormal polymerization? If you remember the earlier slide, I did studies on the pure form of tubulin, and so I knew a lot about it. And what we knew at that time was that metals can cause tubulin to abnormally polymerize. And so we did a study, because at that time there were other people saying the cause of Alzheimer's disease was aluminum, and that was widely accepted. At that time, it was considered the most likely thing, and women were throwing away their aluminum pots and pans that they cooked in because they thought the aluminum coming off the pots and pans was probably causing Alzheimer's disease. And so this is a study that we did at different concentrations, and we looked, zinc can do it, copper can do it, cadmium can do it, mercury can do it. Mercury does it lower concentration than anything else. Can block, as you go this direction, block the binding and make the tubulin. So heavy metals will cause tubulin to abnormally polymerize. But a test tube is not your brain. And in the test, if you know in your body, you have a lot of citrate, which is a chelator. You use it to keep blood from coagulating, binds calcium. It binds all the things. So there are chelators in your body. And so what we did is we thought we'd check that and make sure we were doing it right. And so we did the photo labeling with the metals, and you can see, as we see, the decrease in photo labeling down here at the bottom as we increase the concentration of mercury, cadmium, copper, and zinc. We did it on a lot of papers, but this is the one I used here. But then if you put in EDTA, all of you know EDTA is a chelator. And this kind of irritates some of the medical doctors that like saying EDTA can be used to treat mercury toxicity, because EDTA did prevent the toxicity of cadmium, copper, and all these other metals. Totally prevented it. Actually, if you put the EDTA in, they prevented the toxicity and actually enhanced the polymerization and the binding of GTP to tubulin. But it didn't do it with mercury. Mercury binds to sulfur groups much tighter, and it doesn't bind to oxygen groups very tight at all. So mercury is the outlier, and that's the reason it's so damn specifically toxic to your brain versus cadmium, copper, and some other metals that we know are toxic. Because the chelators, like citrate, etc., oxygen-binding compounds do not bind mercury. They can bind it, but they don't keep it. They don't bind it nearly as tight as it would bind to the sulfur groups in tubulin, where it binds when it causes this problem. So we published this. And I wasn't alone. If you look at this thing here, because some of you say, well, he's trying to exaggerate this, but this was published in 1984 in Toxicology and Applied Pharmacology. I got another one in the Canadian Journal of Biochemistry and Cell Biology. And if you look at this, what they say, 1 millimolar EGTA, which is a form, an analog of EDTA, prevented inhibition by copper and cadmium up to 700 micromolar, but not prevent inhibition by mercury or methylmercury at all. Same thing down here. So what you see is that mercury is an unusual thing. If you're going to chelate, you're not going to do it with oxygen compounds like EDTA or citrate. And so here's the study that proved all this. We put in 100 micromolar EDTA, and again repeated the study with mercury, cadmium, copper, and zinc. And what you can see is that actually the presence of EDTA made the mercury more toxic in the brain. It enhanced the prevention of the full labeling of tubulin. So it's just not going to work. And I'm not against EDTA chelation, by the way. I mean, I think in lead in certain areas it works really well. But what we found here was this is something that's very unusual. And so if you talk about the specific interaction, that tubulin is inhibited in binding GTP in Alzheimer's brain, what can cause that? What could possibly come in there and cause that? And so I published this, and it got the attention of some people. It got me invited to this conference for the first time. And this is the one that the American Dental Association hates the most. If you take 2 Alzheimer's patients, you say Alzheimer's 1, Alzheimer's 2, you can see that the tubulin is not photolabeling. And here's the one with 0 of anything in it, and here's 0, and if you increase the mercury concentration, as we see down here, you see that you make this normal brain look like an AD brain. And this is the way I do research. You know, if you're going to say something causes something, you go back and you can check all of the things that people use to say this is how you define Alzheimer's. Alzheimer's disease is identified as a person who dies, who's demented, who has excess neurofibrillary tangles, excess beta amyloid, and hyperphosphorylation of tau. Not my identification. That's from general neurology and what they say. And we can mimic all of those. We can cause all of those to happen with mercury. And it gets a problem. They say, well, you can't correlate the amount of mercury in the human body. And you listen to Dr. Hughes talk, and which I liked a lot. The NHANES is fine. But if you just look and say, we're going to look just at the mercury toxicity, well, you can't do that. I mean, you can do that, but it's not valid because there's synergistic toxicities. And, for example, in one study we had that was published in 1974, what they showed is if you give mercury, a lethal form of mercury, amount of mercury that will kill 1 of 100 rats. It would kill 1 of 100 rats. If you do the same thing with lead, you can find the amount that would kill 1 of 100 rats. If you mix those two together, or even dilute it, you kill 100 of 100 rats. I mean, so 1 plus 1 doesn't equal 2, doesn't even equal 1/2, it equals 100. That's called synergistic toxicity, which I can explain to anybody that wants to know, be bored for an hour and a half talking science. But so what we're doing here, we did this one to show people the difference because zinc was found to be elevated. And here's the zinc toxicity. You probably can't always see it. Zinc, you have to go really high, up to 20 micromolar, to have it infect tubulin in the presence of anything. But if you take mercury alone, that's the red line, it goes down. If you take mercury with a little bit of zinc that's not toxic, 10 micromolar, it drops it down to here. And if you put in 20, so mercury plus zinc is a hell of a lot more toxic. So if you're out there, a normal person, all of us would have a different level of zinc, cadmium, iron, and other things. Every metal causes the synergistic toxicity with mercury. We didn't test a metal that did not have this effect. So in other words, you cannot just look at mercury toxicity in 100 humans and say, well, it didn't do this, because it'll go up and down. You'll see an effect that will not fit with the concentration. So there's a lot of problems with just taking gross data from 5,000 people and looking at it and saying, this shows this. He did show that it does increase the disease state of these people because it does make people more toxic. And so the question that came back to me right away was, that's in a test tube. You're taking human brain, grinding it up, and measuring the effect of mercury on tissue like that. So we did rats. We took rats and exposed them to mercury vapor. Did this with people at Calgary, the people that did the movie. And what we saw, that mercury vapor, if you put rats in a chamber and expose them to mercury vapor, they breathe the mercury vapor, and the mercury builds up in their body and in their brain. And we're going to show you this study. And here's the study, and what you're seeing here, if you follow the little cursor here, when we start out in a control rat, this is the photolabeling of tubulin, and here is the mercury brain concentration. So you're getting good photolabeling, very little brain mercury. As the brain mercury goes up from here to here, the photolabeling doesn't change because you have protection in your brain called glutathione that binds up the first little ballast. But the next time, if you go from the mercury dosage from here to here and go up higher with a little bit longer exposure, the tubulin photolabeling drops from here down to here, and that kind of mimics what you see in Alzheimer's disease on the ratio. So what this says is that an animal can breathe mercury vapor, and you can induce an Alzheimer's type brain with regard to the abnormal biochemistry, with regard to tubulin polymerization, with mercury and only mercury. You can't breathe any other metal in. I mean, it's just kind of impossible to do that. But we're walking around with amalgam fillings in our house, in our mouth, some of you with 10, 15 amalgam fillings. A person very dear to me had 15 when I saw this data. And that person lost all of her amalgam fillings in about 6 months. So there's no doubt. And so we looked at the full labeling. Decreased NBMI interactions was not due to the tubulin disappearing. It was there. We looked at the immunostaining. The tubulin didn't disappear. It just became non-functional. So what mercury does, it goes in the body, even though the proteins are there and they look normal, on a SDS polyacrylamide gel, everything that you can do to analyze a protein looks normal, but the mercury binds to it and prevents it from functioning properly. And this is the paper I would suggest all of you go look at this, the degenerating neurons into neurofibrillary tangles by treatment of nanomolar levels of mercury. This was done at Calgary. It's called the Calgary movie, a very well done movie. And what they did is they took neurons and they added mercury, and only mercury could do the same thing that we saw that causes the formation of neurofibrillary tangles. That is the major diagnostic hallmark of Alzheimer's disease. Anybody with two neurons left to rub together would look at that and say, this means you should not be putting mercury in people's mouths that might be susceptible to developing Alzheimer's disease. It would at least be an exacerbating factor, if not a causal factor. Unfortunately, most of the people in our FDA that addressed this issue only had one neuron left. Okay. Now here's a paper that was published in 2010, and it says decreased clearance from the central nervous system of beta amyloid in Alzheimer's disease. In other words, in Alzheimer's disease, they're not making more beta amyloid. They just can't get rid of it. They can't get rid of the excess. A good hint of what might be happening. And this, it totally fits in with what we're saying. So this came from Korea, Dr. Song and Cho. I'd like to meet these guys sometime because they did a very elegant piece of work. Because beta amyloid elevation is the second major diagnostic test for Alzheimer's disease. But you don't want to attack beta amyloid because beta amyloid is the target of all the pharmaceutical companies that want to make a ton of money off of people dying of Alzheimer's disease by giving them something that might regulate it or do something to it. And they have. They've made antibodies that bind beta amyloid. And they killed all the patients they treated with it. Not all of them, but enough of them that they quit. So, but what these people did is they took mercury at different concentrations, and you look at the mercury level, methyl mercury, fish, or then all of them, and you see a big increase in beta amyloid levels with the exposure to mercury. So that fits into the thing. That's what you would expect to see. This is just a graph of the same thing. So you can read these papers and the references here. So beta amyloid is increased by the presence of 10 times 10 to the minus 9th molar mercury in the brain, or in this tissue, these cells that they studied. That's a lot lower than what's in your brain. I mean, people sitting here right now, most of you will have more than that level of mercury in there. And so they had to figure out how it happened. And see, there's an enzyme called nephrolysine. It's an amyloid-degrading protease put in the body to get rid of beta amyloid. And as you increase mercury coming across, this is the expression of nephrolysine, it decreases. So mercury prevents the production of the protease that chews up beta amyloid, allowing it to build up in your brain, and causes the second major diagnostic hallmark of Alzheimer's disease. I mean, this is not that. I mean, anybody can repeat this data, and I haven't heard anybody say it's not there. And while the I can't keep track. While the nephrolysine, you can see, is going down in quantity, if you go up here and look at the amyloid precursor protein, you can see it's increasing because it's not being chewed up. And so that's a process that explains the earlier report saying, why in the hell does beta amyloid build up in the brain? It's not the cause of the disease. This is the biggest joke that's been played on the American public. We have funded hundreds of billions, probably billions of dollars, without a doubt, to look at beta amyloid as, quote, the cause. Why? Because some half-baked professor at Harvard said this is the cause of Alzheimer's disease. It's not. It's the result of Alzheimer's disease. The cause is something like mercury and maybe other things. I wouldn't say mercury is the only thing, but it's the cause of the synergistic toxicity and the fact that we live in a society where heavy metal, zinc, copper, iron, everything is going wild, but mercury especially is going wild on these people. So if we look at this, what we can say is that mercury causes the production of neurofibrillary tangles, the abnormal polymerization of tubulin seen in Alzheimer's disease, and the buildup of beta amyloid. This is something that we published sometime with creatine kinase is over 95% inhibited in Alzheimer's disease brain. Creatine kinase, if it's in your heart, if you inhibit it 95%, you drop dead in a second. But in Alzheimer's disease, it's a slow process. As the mercury builds up, it inhibits it. It's over 95% inhibited in AD brain, and we showed that it's inhibited by mercury. And we published this, and it has a very reactive sulfur group in the active site, which fits in with the science of this. And this is creatine kinase exposed to water, where we took a dental amalgam in a tooth that some dentist pulled out of some poor person and gave to me that used for testing. So I would take this tooth with an amalgam. I used it I don't know how many times and how many experiments. You drop it into 2 mL of distilled water that's totally non-toxic, and then you measure the time it sits in there, and then you say, what effect is the mercury? Can this amalgam filling in a tooth cause the water to become toxic to the brain? Well, it does, especially for creatine kinase, which is one of the more sensitive enzymes. But here's creatine kinase, how much creatine you would make, creatine phosphate with the creatine kinase, and here would be if you had it in added water that was exposed to that amalgam for 15 minutes, 1 hour, and 24 hours. So what this says is that dental amalgams can make water, like your saliva, if you're sucking on it and you're swallowing it, toxic to creatine kinase in your body. And you can ask any medical doctor, creatine kinase is a fundamentally incredibly important protein in your body. And it has to be active for you to be. If you don't have creatine kinase, you don't have any energy. And this is one of the reasons why you'll see people who are mercury toxic are very low. I mean, they suffer from chronic fatigue because they cannot transfer. Creatine kinase is used to transfer the energy from ATP to certain other aspects where it can be used effectively to keep you energetic. So here's some other reports on mercury and Alzheimer's disease. This one here was published by Oliveira in 2000, and it says, agreed that neuroblastoma cells to 10 to the minus 9th molar mercury increases. Tau phosphorylation, hyperphosphorylation is a hallmark, diagnostic hallmark of Alzheimer's disease, and the secretion of beta amyloid. And then we can go down there, exposure of neurons to 10 to minus 7, 10 minus 10 molar mercury causes the formation of neurofibrillary tangles. So right there at that time, that was the 3 major diagnostic hallmarks of Alzheimer's disease that you could induce by putting mercury in, and yet you cannot. When I applied to the NIH to study this, the effect of mercury on these factors with regard to Alzheimer's disease, they would not fund it. I had been funded for 25 years plus years doing this type of study. But when I started working on mercury and correlating mercury toxicity to Alzheimer's disease, I couldn't get a grant. And if it hadn't been for foundations and people in this academy that helped me find people, my research would have come to an end. We do not have a government that's friendly to people who want to say that mercury is toxic, especially mercury that's put in your body by dentists or medical people. They won't admit that. The FDA approved those, so they're definitely an antagonistic group of people with regard to that. And then we come down to why this works. This is the study from Korea. Levels of mercury inhibit nephrolysin that breaks down beta amyloid. If you want to do it, get the mercury out of your body. But there's no money there. Don't you guys understand? There's no money. I mean, Eli Lilly, Merck, and these guys aren't going to make any money if you just take mercury out of the body. They want to give you a drug that you take every day, and they can sell it to you for a huge amount of money. And that's what drives, and that's what I'm saying, one scientist view. I've been in this now fighting to get something to go and take the mercury out of your body. You'll find there is a lot of pushback on that. They do not want people to become healthy and wise and not affected by mercury and other heavy metals. I have very little faith in the FDA doing anything in this regard. Now, look, there are good people in the FDA, and I don't want to go, cover them all with the same brush. But the people who get appointed on the top are political hacks, and they do things for political hack reasons, and we've seen it time after time in this academy where we've gone to the FDA, talked to their top scientists, and hell, they agree with us. They say, Dr. Hell, you're preaching to the choir. We know it's toxic, but we can't do anything about it. And I remember asking the guy, Rich Fisher and Mike Fleming took me there, and with Dr. Cho, and I said, well, wait a minute, you're the FDA, the buck stops here. He says, no, I can't. And this is the top scientist at the FDA. He said, I first have to get the permissions from the Office of, it started with an M, Management and Budget is what it is. But he said, I can't tell you. And so when we put in our report about the science, it went to the Office of Management and Budget, who are not scientists, they're money people, and it got torn apart. I mean, it's redacted, everything taken out of it. They couldn't argue the science. There is no science in existence, and I look hard because I don't like being wrong. There's no science at all that shows that amalgams are safe. And look, the United Nations has the Minamata Bay Treaty. I suggest you go to your computers, Google Minamata Bay, and what they're doing. The rest of the world is phasing out amalgam fillings. The only people I heard argue against that, because I was there and testified to get rid of them, the only people that argued in their favor were people from the American Dental Association and the Canadian Dental Association. So you all are guilty. I mean, they're the ones that say this. So look at this. Here's a comparison of the 80 pathological hallmarks and those induced by mercury. The check marks was induced by mercury. Abnormal tubulin GTP binding. There's no doubt that that's a major function. Check. Neurofibrillary tangles form. Check. Beta amyloid levels increase. Check. Decreased creatine kinase activity. Check. Decreased glutamine synthetase, which I didn't talk to, but because I only got so much time. So you have all these, and I think the word is ipso facto. With these proof, you have to say, this is how you define Alzheimer's disease. It has these abnormalities. Mercury causes Alzheimer's disease. It may not be the only thing that causes Alzheimer's disease, but anybody who says it doesn't and cannot is either a liar, a crook, or a fool. And I won't back down from that one bit. And if you look at how much money we spend on medicine in the United States, we really ought to be telling the CDC and the FDA, get the hell out. Just wipe them out. You don't want those people there doing. But we should support somebody that would go to everybody that's 35 or whatever age and take their amalgams out. If you don't want them to have serious disease, stop the toxicity, especially that induced by medical personnel, such as dentists and medical doctors, and take the damn mercury out of vaccines. Okay. That's a little bit more, because I'm going to beat on the Alzheimer's a little bit more, but right now I want a temporary change of topic alert, because I don't want to not be able to get this in, and that's what I was worried about, because I start talking, sometimes I talk too much. But we have some brain and liver iron toxicity studies in HD67D genetically modified rats that slowly retain Fe in their body, iron. Free iron is very toxic, by the way. Iron is bound, you need, but free iron is very toxic. And then induce dementia and loss of limb function. Treatment with NBMI, if you give these rats that first become demented, I'm not good at measuring the dementia in rats, but other people are. And this is how long it'll sit in a bright light or something like that. But they say they become demented, but they can't walk. I mean, I can measure that. If they can't walk, they lose the functionality of their limbs. And this is a model for neurological diseases such as Parkinson's. If we take those rats that we know are going to die within a week or two and become non-functional, and we give them NBMI, none of this happens. They don't become demented, and they don't die. And they don't lose the use of their limbs. And if we do a post-mortem analysis, if we show that the elevated iron gets in the brain, induces oxidative stress, such as in the brain, when you have mercury go in, what happens to the iron in your brain? I think one of the previous speaker, Stephen, talked about that. It knocks iron off of its binding sites, and that iron now can participate in a Fenton reaction and generate hydroxy radicals, which causes the breakdown of the brain. So we have that. So we did that, and this is one on tau phosphorylation. The iron-induced stress does that. But here's the data from this, and so I'll not spend a long time on it, but I want to show you something, because this tells you something that's important. This is the the iron toxic rat is 18.6. If you treat them with NBMI, their iron level dropped down to 16, and the control that is not genetically modified was 15. So you see an improvement. Now, if you look at copper doesn't change, zinc doesn't change, and manganese doesn't change. So then we pull out their brains and we look at oxidative stress markers, and you can see that oxidative stress is going up in the rat that has the high iron. If you give them NBMI, the oxidative stress is totally eliminated. Because even though you see that little bit of excess iron in the brain, that iron is bound by NBMI, and NBMI does not prevent toxicity by causing the elimination of the metal. It prevents toxicity by binding the metal into an inert, non-toxic, non-reactive form that cannot induce oxidative stress. And the other interesting thing on this is you look at the liver iron. The rat that wasn't treated was up here, and with the NBMI, it comes down to 189, from 864 down to 189, and a control rat is 152. So this means that this compound is going to be effective, very likely. I mean, I would predict that, and that's my prediction. It's not proved by the FDA. You know, you have to pay them a lot of money and do a lot of things to get them to approve your stuff. But thalassemia, iron overload diseases, hyperchronicity, there are several things. This compound will tie up the iron throughout your body and render it non-toxic. And so that's something we're doing. And we're doing that because it's very difficult to prove that mercury is the brain iron study demonstrates that NBMI can cross the blood-brain barrier and chelate a toxic metal and eliminate the damaging oxidative stress induced by this metal. In this case, it's iron. But it would bind mercury even tighter. So this should be something that would be useful to treat people that are in the early stages of Alzheimer's disease if I can never get it through the FDA or the EMA at this time. It makes sense. This compound has absolutely no toxic side effects at the level we use for treating heavy metal toxicity. It's very good. This is what I've been working on, and the reason we went to iron is because nobody wants to touch mercury. Mercury is considered taboo, mainly because of the actions of our government. And you saw it in the 2006 white paper. That's not that long ago. Dental amalgams are safe for everybody. I mean, they don't leak mercury. The mercury they leak is a vitamin. Okay. Here's the thing. Now, you know we have a real problem with lead intoxication in the United States. And I'll talk to you about the compound. NBMI binds all of the different heavy metals very tightly and will precipitate them out of solution. And that's because of its structure and the way we designed it. But we did some lead intoxicated studies with rats because nobody wants to fund the mercury research, but they are somewhat interested in the rats. And here's the lead increases the white blood cells and lymphocytes in the rats. If you take rats with nothing, and then you look at the white blood, here's the control. Initial PB level is right here. I mean, the white cells go from here up to here with the thing. And then if you treat them with a vehicle, if you treat them with NBMI, they drop from here to here very fast. The same thing with lymphocytes. They go up very high with lead. If you allow the lead to be kicked out of the body naturally, it drops down. But if you treat with NBMI, it goes down to 2.9 very quickly. So what we have here is proof that NBMI will work on iron and lead as well as mercury, which doesn't surprise someone like me. I mean, that's the chemistry of the compound. So you can read that as well as I can. But what we're saying here is that it works out. Now we're going to go back to mercury and Alzheimer's disease. This was something, when I talked in 1989, I had no idea there was mercury in dental amalgams that would come out. I knew there was mercury in there. I know what an amalgam was. But I couldn't believe that the FDA or anybody with good sense would allow you to put something in someone's mouth that would leak mercury. And so I was shown that and surprised, and I thought, well, this would be easy. We'll just show this to the FDA. You know, I can measure mercury coming off an amalgam. That's high school chemistry stuff. It's not hard to show that the mercury is toxic. What we have is we have a very corrupt medical bureaucracy, not only in the United States, but in Europe also, that will not do anything that interferes with physicians and dentists making money. They won't. That is what I'm saying, a single scientist perception. It's not the science. The science has been published time and time again by hundreds of good scientists and totally ignored by the government agencies. You should never let a politician make a decision about the toxicity of a compound or the safety of a compound, because they'll go with the money every time. And now, for example, here's the study, that should get everyone. This was funded by the NIH and done on orphans in Lisbon, Portugal, which concluded that amalgams were safe for use in children. They put amalgams in children. This was done from the University of Washington, And I think those people should know better, and they're probably embarrassed that this has come out. But when we read this, we knew it couldn't be right, and we pushed on this study. And what you see at boys, this is the year one, or it's zero thing when they start out when they were 7. When they get out here, they're 14. And if you look at the mercury coming out, and those that got amalgam fillings versus composite, the white lines, you see the boys go up, they start releasing mercury in their urine, and then all of a sudden that mercury goes away. It goes down to at the end of it, there's no more mercury in their urine. In other words, they're breathing in the same vapor as the girls over here. The girls did the same thing, but the girls go up and level off. And they're still excreting the mercury into their urine. And where did that mercury go from on the boys? Well, boys are 4 times more likely to become autistic, 10 times more likely to become Asperger's, well, that used to be, or to have neurological problems. And we did the studies in the laboratory, and I presented to this group before. If you put testosterone in with neurons in culture and add mercury, they're all dead in 3 hours. If you don't put the mercury in, they'll live out for 24 hours. If you put in estrogen, the female hormone, they'll live even longer. So the female hormone protects. So when you go and you look at 5,000 people, you won't know their sex. If you say, is the mercury having an effect? And we pushed this quite a bit down the line. And what we found is that there are genetic differences with the boys also. Some of the boys, if they have a CPOX gene allele that renders them susceptible to mercury, they're going to be affected and be very sick very quick, much quicker than the other boys. And if we look, okay, so we back up and say, there's no doubt amalgams are bad to these kids. And if we look at the total mercury by anatomic site and number of occlusal amalgam fillings, this was done on in Turkey, on Turkey medical soldiers, and it was published in the American Journal of Forensic Medical Pathology in 2006. And they look at the scale. If they had 0 to 3 amalgam fillings, are here, but now, and this is 0.025 about, let's say, in this region. And if you go up here, if you put in 4 to 12, it goes up to above 0.05. And if you go to greater than 12, you're up here where it's above 0.1 micromoles. And so there's no doubt, I mean, this is a log scale. There's no doubt that increasing amalgam fillings increases the amount of mercury in your body and increases your toxicity and the effect that you'll have. So they say, does mercury come off of amalgam fillings? We took an amalgam filling, the one I told you about before, dropped it in distilled water, took aliquots of that after being soaked for a period of time, and added it to normal brain. And an amalgam filling makes a distilled water solution so toxic that it can totally prevent the GTP binding to tubulin, which you see in Alzheimer's disease, in 1 to 2 hours of soaking. And so anyone that says an amalgam filling, that there's no proof, the FDA knew about this data. They just chose to ignore it. And not only this data, but the data from probably 100 other scientists. So, okay, here's the last one, and then I'll shut up. The Apoe genotype and risk for Alzheimer's disease. It was done by a person at Harvard by name of Roses. And what they said was that ApoE4, if you have 2 copies of ApoE4, you get one from your mom and one from your dad. If you're ApoE4, you're going to get Alzheimer's probably before you're 60 years old. If you have 2, which is the one type, you won't get Alzheimer's disease if you live to be 100. So it's very protective. And if you look at that, and this was very important to me, if you look at the type, if you get a 2 from your mom and a 2 from your dad, you'll never get Alzheimer's disease. If you get a 4 from your mom and a 4 from your dad, you're 2% of the population. Sure, you're going to get Alzheimer's before you're 70 years of age. So there's no doubt that this tied in to Alzheimer's disease susceptibility. And so the chemist in me, when I saw that, and we published that first, a long time ago. And if you look at the difference, what's the difference? ApoE2 has 2 cysteines on the surface. This is the only change. In the ApoE3, which binds can bind 1 mercury, one cysteine is converted to an arginine, and the ApoE4, you've lost both your cysteines that can excrete mercury. Now, what's important to know is that ApoE4 protein is made in the brain. It's a housekeeping protein. It carries oxidized lipids and cholesterol out of your brain, across the blood-brain barrier, into the blood, and it gets cleared by the liver. So it's taking junk out of your brain. And so while it's taking out the oxidized cholesterol, it can be taking out mercury too. So it's a counter current. It's going in the opposite direction that mercury would be going in. And If you look at this, the relationship, and we published this in Metal Ions and Biological Systems in 1996. If you have 2,2, you have 4 of these sulfurs you can take out, you're going to get mercury, you're going to become diminished at greater than 90. If you go down through here to 2 and 1, if you go down where you have none, like a 4,4, you're going to get Alzheimer's disease before you're 70. And so that is not a diagnostic hallmark, but that is a marker for Alzheimer's disease risk that is explained and can be explained by the fact that the ApoE4 protein that protects you takes the mercury out of your brain and prevents it from getting in there too. So if we look at the factors implicating mercury in the etiology of Alzheimer's disease, you can read this as well as I can. There's a whole load of them, and there's more than this, but this is the ones that I know firsthand that they're correct. So what's the problem with us not getting some response from the government on mercury? It's not the science. There's more science than this and done by people that are in more prestigious locations than I am. The problem is we have a dishonest, dysfunctional Food and Drug Administration and Health Service Department in the United States. You will go to NIH and ask them, how much money have you spent trying to develop a chelator to remove mercury from the brain or from the body? They don't spend, I don't think they spend any money on it at all. I've never seen any publication funded by NIH on the development of a new chelator to prevent heavy metal toxicity. And that is one of the major things that I did, because you can't out-PR these SOBs. I mean, they can find people that will sit there and write the most negative character assassinations, and they've done it on many people in this thing. If you're a dentist and you say, I think mercury's toxic, what happens to you in your hometown? I mean, with your local dental board. You know, I mean, I know I've testified for a number of you. They can't fight you scientifically. They run if you say, let's talk science. But if they can just kill your character, assassinate your character, they'll do it in a heartbeat. So the real thing, and you can read this, it's all on the slides here, that we have to do is we have to get politically active, because we have a corrupt medical authority in the United States. That's the only thing you can say about. At the very top, they're corrupt. And don't look at it any other way, and that doesn't help my company or me at all to say that. But that's exactly what they are. They're not even concerned. I sometimes think they don't even want people to get well. I mean, if you take away the beta amyloid thing, what are the people in the big pharma companies going to do, and what drugs are they going to come up with to make you make money? I mean, you've got to fund them. I mean, that's the major concern they have, is how do we fund these people? So I'll quit right there, okay? And there's no help from the government. It is my opinion that presenting causation data to regulatory agents was not an effective method to eliminate the toxic effects of mercury and other metals to our citizens. And that's the reason I decided to make NBMI. I can't out-PR them, and you can't convince them, because I think they're crooks. I mean, I don't think they're honest people. And I would say that in front of Congress. I have said in front of Congress that, as a matter of fact. They're just not honest. And we accept it. And the real problem is the citizens do this, and it's your mom and your dad, your grandparents that are going to get this disease if we don't stop and start doing something about it. They'll let this go on forever. And so when you talk about some of the issues that come down with mercury toxicity, etc., this is what I think the major problem is. Okay, I'm done.